in the Americas. For example, the Western is widely distributed through the Americas, from Canada to Argentina. Now, those diseases are important because of the severe lesions that they cause. They can cause disease not only in animals, in equine, but also in the humans. Up to 33% mortality in severe cases of Eastern equine encephalitis. In fact, the other diseases, they cause up to 80 and 90% mortality in some cases. The main reservoir is thought to be birds. Mosquitoes take the virus from birds and infect horses as well as humans. Now, this disease is called equine encephalitis virus. However, the progressive form of this disease causes encephalomyelitis. So it not only involves the brain, but also involves the spinal cord. Hence the importance of this, because the symptoms as a result of encephalitis is different from the symptom as a result of encephalomyelitis. When it comes to encephalitis, for example, the signs are impaired vision, photophobia, inability to swallow, circling, yawning, and grinding of teeth. But when it comes to spinal cord, then become impairment of movement, like ataxia, recumbency, circling, pushing the head against fences, against a fence, etc. So all of these symptoms are important also for diagnostic purposes from other diseases that cause encephalitis instead of encephalomyelitis. When it comes to mortality, I said it's pretty high. In Eastern, it's 50 to 90%. In Western, 0 to 40%. And in Venezuelan, up to 80% mortality. There is a characterization of some of those horses, they're called sometimes dummies, implying that they have they're dull, they have a dementia, they cannot remember the instructions given to them, and that's because of the obviously encephalitis. Okay, when it comes to the pathogenesis, so basically the virus enters the blood circulation following a mosquito bite. It infects the dendritic cell. The dendritic cells take it to the lymph nodes, and then the virus in the lymph node replicates, causing primary viremia. Following primary viremia, there is a secondary viremia, because now if you want, the number of viral particles have increased dramatically. So it enters the blood again, and from the blood it can now reach in large number the brain and the CNS in general, in the progressive form. What are the characteristic histological lesions? Neuronal necrosis and neuronophagia. Basically, is necrosis, and neuronophagia. Phagia refer to the monocyte. T refer to macrophages. In this case, in the brain, there are the microglia cells. Microglia cells, which are recruited to the site of infection around the blood vessels, and neurons that are infected. Hence, it's called the neuronophagia. So there is necrosis and neuronophagia. When it comes to diagnosis, one can aim for the antibodies and can aim for the viral antigen. In acute phases, that's the beginning of the infection, one can do an IgM ELISA and that's helpful when it comes to screening of several equine. Because the antibodies are present not only in the blood circulation but also in the cerebrospinal fluid, one can take the cerebrospinal fluid and look for antibodies in the cerebrospinal fluid against this particular virus. Because this is an RNA virus, other techniques are TPCR, immunofluorescence, or immunohistochemistry, all of them in this case looking for the presence of a virus or a viral antigen. Okay, so now comes the important part, differential diagnosis, because there are a number of diseases that can cause encephalitis and encephalomyelitis. First of all, the presence of fever with neurological sign occurrence in the fall is indicative or is a characteristic, if you want, of some of those diseases. So those diseases, some of them, they occur in the fall, like West Nile occur in the fall. And that's basically because of the transmission due to mosquito presence. So West Nile, it's a flavivirus which is causing which causes encephalomyelitis. Do we remember in the bovine another flavivirus? Bovine viral diarrhea. Okay, so bovine viral diarrhea comes from has another genus which is different from West Nile and St. Louis encephalitis. Bovine viral diarrhea is not transmitted by mosquitoes, okay? It's a respiratory disease. Or mainly, if you want, the entrance of the virus is through the respiratory route. Whereas in the case of the West Nile and equine encephalitis viruses, they're transmitted by mosquitoes. So another disease, St. Louis. St. Louis is also present in the U.S. It's endemic in a number of in several parts in the U.S. But usually horses, they do not develop the disease. They act as a reservoir of equine encephalitis viruses. Equine protozoal myelo encephalitis. That's caused by Sarcocystis neurona. Have you had this in the first year? Protozoa. It's a protozoa. Basically, sarco refers to what? Muscles, exactly. And cystis refer to muscles, to cyst in muscles. But when it comes to Sarcocystis neurona, in this case, the protozoa can enter the brain and can affect the neurons. So it causes neurological signs, both cranial and spinal, meaning it causes encephalomyelitis. There is, in Zuku review questions, this disease come in the differential diagnosis, and for reason that I did not find out is that there are localized strips of sweating. Seems to be diagnostic for equine protozoal myeloencephalitis. So I don't know what it refers to or what's the cause of localized strips of sweating. But I guess fever is present in other also diseases which causes encephalitis. The other one is poisoning by yellow star, eating yellow star flower. This happens when horses do not find food and they target areas that are not really supposed to be. They're not supposed to be in those areas. If they eat it for a prolonged period of time, the poison from these flowers can cause neurological symptoms. So that has to be also differentiated from it. However, the clinical history in this case is important because it will be indicative of those horses, grazing, if you want, on those flowers or eating these flowers. It should be differentiated from herpes 1 and 4, particularly herpes 1, although that's not necessarily in the 4. Herpes 1 can occur at any time. When it comes to immunity, it's probably lifelong immunity. There are inactivated vaccines for each of those viruses. The horses are vaccinated annually, and the foals are vaccinated. And when it comes to control, obviously, if you can control the mosquitoes, the larvae, and the movement of horses, then it will help in this regard. But the vaccines seems to work particularly well. And the example is not only equine encephalitis viruses, Eastern, Western, and Venezuelan, but also West Nile virus vaccines are pretty good in that respect. So West Nile virus is another flavivirus, then the genus flavivirus. It's an RNA virus like the others, and it's arthropod borne. Again, it's very similar to the other diseases that we just talked about. The reservoir is mainly white birds. The mosquito can spread the infection, transmit the infection to humans and horses. In the US, it can cause high mortality in birds and encephalitis in both humans and horses. Now, when it comes to neurological signs, these are signs as a result of encephalomalacia or encephalitis, which include depression, abnormality in gait, head tremors, muscle fasciculation, etc. So, various nervous or neurological signs which can occur as a result of the infection. Okay, the lesions basically in the brain and spinal cord, which in this case the lesions are lymphocytic encephalomyelitis. So this includes the recruitment of lymphocytes to the site of the infection, as well necrosis, as well as necrosis. The diagnostic test the same as in the other diseases the Eastern, Western, and Venezuela, which include the IgM during the acute stage of the disease, and virus neutralization assay looking for antibodies, RT-PCR, immunohistochemistry, and cell culture. A differential diagnosis, it should be differentiated from the diseases that we talked about. The Eastern, Western Venezuelan, St. Louis, the Sarcocystis neurona, and yellow star thistle causing diseases. Vaccines are available also, attenuated and live recombinant. Particularly in the case of the equine, the DNA vaccine seems to be working pretty well, and DNA vaccine can overcome maternal antibody issue. But there are other vaccines available, like canary pox. So those are, remember you can use another virus that usually does not cause disease in the host you're giving the vaccine. In the case of canary pox, it does not cause any symptoms or lesions in horses. So it can be used to carry genes that encodes for proteins, and also proteins when they are injected in horses that produce a good immune response, that can protect against West Nile virus infection. And last one, familiar one, that's the papilloma in horses, and that's called equine sarcoid. And we know the difference between the bovine papilloma and the equine sarcoid. And in case of equine sarcoid, if you try to remove them, they reappear. And the reason for this is because the immune system is non-existent against equine sarcoid. Against bovine papilloma, it seems to be effective. The immune response is effective. It can control the virus. And if they disappear, usually they do not reappear. But in the case of equine sarcoid, they do. So those are growth, but they're not malignant growth, which is also important. The different type, there is like the bovine papillomas, the wart-like and the fibroblastic, the mixed and flat types. So all of these can be present, and now the causative agent, if you want, of equine sarcoid is not well known. However, it is believed that bovine papilloma viruses are the cause of that infection. The reason for this is because they've been isolated from those lesions. Now, as I said, the horses do not develop an antibody response against equine sarcoid. Some attempted instead of inducing antibodies, inducing cell-mediated immunity, which makes sense in terms of dealing with growth or in this case benign growth. So how do you boost cell-mediated immunity? You can boost it with a number of using immunopotentiators like adjuvants. Those adjuvants, in this case, if you remember, I mentioned BCG, Mycobacterium tuberculosis. It can act as an adjuvant. It can boost cell-mediated immunity. So if BCG is injected in those animals, it seems to result, well, based on research, it seems to cure those animals from sarcoid. But also others can be attempted, like surgery, laser surgery, radiation, drugs, which have variable success, because in places where difficult to reach, it's very difficult to remove if you want this growth. Okay, any questions? Do you have exam today?
We had one.
You had one. Okay. How did you do? Good. Excellent.